Evolution has so many insurmountable problems and so many false predictions. And here today, we're going to be talking about one of the false predictions of evolution. Here with me is Dr. Cornelius Hunter. He's a biophysicist, and we're going to be talking about the problem of protein evolution. Proteins were supposed to have evolved early on in life, and we're going to find out that that's a big problem. Welcome, Dr. Hunter.
Thank you, Rebekah. Glad to be here. Do you want to show a cool video to begin with?
Yes. So, we just want to show some of the molecular machines that are formed by proteins. Proteins are little molecular machines, and sometimes they're formed together and make giant complexes that do amazing things in our cells. So I just wanted to give you an idea about what we're talking about here. So here you've got a kinesin transporting things around in the cell. This is just one of so many molecular machines. Here's ATP synthase, absolutely necessary for life. So these are proteins coming together to form these amazing molecular machines that do things that are absolutely necessary in the cell. Here's a strand of RNA that's being cut. The introns are being cut out of it. This is a spliceosome coming, and these are amazing things that must happen in the cell. In fact, the most simple cell in all of life has at least 500 different types of protein machines. So these are absolutely necessary. Here's a ribosome, which is constructing proteins. And so these are just fundamental molecular processes. These have to be able to evolve. So, Dr. Hunter, I'll let you take it from there.
It takes proteins to make proteins, as we just saw in the ribosome. Yeah, so what are proteins exactly? So proteins are made of amino acids. So recall that there are hydrocarbons in our cells. There are genetic material, DNA and RNA, and there are proteins. So the hydrocarbons are made of carbon and hydrogen, and the DNA, RNA are made of nucleotides. So proteins are made of amino acids. Now these hydrocarbons, they're made of, these carbons are lined up more or less in a line. Now there can be little jags in the line if you have double bonds and such. So it's not always a perfect straight line. But then there can be forks. So a carbon can fork off into two sequences or two lines of carbon atoms. So it's branched. So that's not the case for the DNA and RNA, which are linear or unbranched, linear, sequence of nucleotides. Same thing with proteins. Proteins are a linear, unbranched sequence of amino acids. So that's a key distinction, unbranched, linear. Now, in the case of the DNA, it easily forms a coil. So everyone's heard of this double helix, so the double coil, double helix, where you have two strands together. And then, of course, it's kind of easily bent, and so a long stretch of DNA can be wound around a protein hub to make a nucleosome and so forth. So DNA can take on a lot of different shapes. RNA also can take on hairpins and so forth. So it's wound around. The amino acids, when you put amino acids together in a line, in a sequence to make a protein, it's a different kind of a structure that you're going to get. And so when you hook these amino acids together, first of all, every amino acid has a backbone. They all have the same backbone. It's only like 4 atoms or something. There's a short little backbone, and then off of the backbone is hanging what's called a side chain, and that's what changes from amino acid to amino acid. So in the backbone, there are 2 carbon atoms. One is called the alpha carbon, the other is called the beta carbon. And so the side chain is hanging off of the alpha carbon. So, okay, a couple of carbon atoms, side chain hanging off of one of the carbon atoms, you have a backbone. When you attach the amino acid to amino acid to amino acid in a long linear unbranched chain, you're forming a big, long backbone. So you can think of a protein as this big, long, serpentine sort of backbone that can change, that can flex, and then side chains hanging off at even intervals. For each amino acid. So and then if you know the shape of the backbone, you pretty much know the protein. The side chains will point in predictable ways. So you can kind of predict where the side chains are pointing. So just knowing the backbone, and if you just know that alpha carbon atom, where that is, then that forms the backbone. That gives you the shape of the backbone. So that's one way to model, a simple way to model proteins is just give me the alpha carbon atoms. So there's one alpha carbon atom per amino acid. So you have a series of these alpha carbons. That gives you the backbone, and you can predict where the side chains are pointing. And if you know the identity of the amino acid, you know what the side chain is. So that's a good approximate way of modeling the structure of a protein, the three-dimensional or tertiary structure of the protein. Just the sequence of amino acids, that's referred to as the primary structure. It doesn't give you the actual positioning or anything. It's just what is the sequence of amino acids. And then when you have these amino acids hooked up together, they'll take on some local shape. And that local shape is the secondary structure. And then the tertiary structure is the overall, if it folds up, it's called a globular protein, if it folds up into a glob. And so a lot of proteins are globular proteins. And so that is the tertiary structure. And then when you have different proteins that come together to work in a molecular machine, which is very often the case, they often work in teams, not in solo. That's the quaternary structure. So like, for example, hemoglobin, that very important machine in our carrying oxygen around in our blood and carbon dioxide and so forth. That's 4 globin proteins, 4 globins put together. So that's the way proteins work in general. I want to say a word about this structure some more. When you have 2 amino acids hooked together, like I said, you have a typical protein could be 100, 200 amino acids hitched together. And so a common analogy is like a train where the train cars are hitched together, or beads on a string. So that's the way the protein, all these amino acids like a beads on a string or train cars. However, the analogy breaks down because you have this serpentine shape, some sort of weird shape to the backbone. And it's a little bit restricted because when the amino acid is hooked up to another amino acid, there's a special kind of bond that forms when those 2 bond together, a chemical bond called a peptide bond. And so if let's say the first one is here, it's frozen, and I'm going to stick it right there. The second one can only be in certain places. It's restricted, okay, but it does have some flexibility, kind of like a chain. You can wiggle it around a little bit, but it can't go in any shape. And so the second one, it can rotate around an axis, so it's kind of like a cone. It's describing a cone. So picture, like a cone, and this second the alpha carbon is going to fall somewhere on the surface of this cone. So the first amino acid is down here. The second one can rotate around this cone. And so there's some interesting little flexibility on where the second one can be, and then the next one after that. And so if you have, a whole bunch of these, you can kind of configure it to form some sort of a structure. One particular setting in this cone is that kind of a tight coil. If you have consecutive amino acids in that at that extreme, it will coil around. And so you'll have a helix. So it's kind of familiar, right? Helices are a common thing in molecular biology. You see the helices in the DNA. You have now helices in proteins. They're called alpha helices, but they're helices. And they vary. This is the most common secondary structure in proteins. It's like 2/3 of the time, practically, proteins are going to take on this helix shape. It can only go so far It's not going to be like super long typically. In most globular proteins, a helix maybe is 10 or 15, 5, 10 or 15 amino acids long. Another shape is where it's stretched. So that's where it's kind of compressed into this helix. Another shape, an opposing shape, is where it's stretched out as much as it can be. And so it's kind of a jagged W shape that goes on and on and on. And that's called a strand. So you can stretch them out into a strand. That's the second most common shape. And so the strand is like 20% of the time or something like that. It takes on a strand shape. Like I said, it's finite in how far it will go, maybe a dozen amino acids or so. So then it'll do a turn of some sort. There'll be some sort of a turn or a coil. It very often will be another strand, and the two strands will be adjacent, and they actually will interact. They'll bond, very weak bonds, hydrogen bonds between the strands. And then you have another one for a third strand or fourth strand. And so it actually can form a sheet. And those are called beta sheets. So it's kind of same thing with the helices. They also can have a turn, come back, and so you can have helices that are next to each other. And those are called bundles, alpha helix bundles. So then you get into these kind of, you're building up the three-dimensional shape by doing this. These are very difficult to predict. What shape is it going to take on? And so it's a very interesting problem. The protein structure prediction problem. Very difficult to predict that. It's closely related to another problem called the protein folding problem. Is you build this, like you showed the ribosome, and you're building this protein, and then when it gets done, or as it's being synthesized, it is folding up. It's going to fold into some sort of shape. So how do proteins fold in the cell, and how do they attain the final shape, the final tertiary structure that they attain? It's very difficult problems. And it took artificial intelligence to really make some breakthroughs. And so in recent years, artificial intelligence was brought to bear on, feed it all the data that we have, all the structures that we know of, and their amino acid sequences, and tell us, how to make this prediction. And it actually worked pretty well. It has some better success than we've had to use artificial intelligence to do this. Of course, the big drawback with artificial intelligence is it's a black box. It doesn't actually tell you what's going on or why it did that. It's just, it absorbs all the rules, all the knowledge that we have, and comes up with good inferences without knowing what the purpose, what the reason was. So it doesn't really help you design a protein. If you want to say, well, I want a protein that looks like this, has this shape, what should my sequence be? That's a hard problem. It's a hard problem, and it doesn't really help that much. There's ways, it's certainly going to help somewhat. So it's very, very interesting stuff. If you could bring up a couple of examples just to show how cool these, so this one is ribonuclease inhibitor. So ribonuclease is an enzyme that will chop up RNA. RNA nuclease inhibitor inhibits that enzyme, and so it protects RNA. Why I like this one is it's cool. It's just a cool shape. So if you look at this, the gray part, the surrounding kind of white gray part, is if you were to draw the protein using every atom, not the hydrogens, but the carbons, the nitrogens, oxygens, and each one kind of forms a little ball. And so you draw a little ball for each one and put them all together. You get this ball, a bunch of balls, like a jumble of balls, together. And it really doesn't tell you very much, does it? You look at it, you give the overall shape. Oftentimes it's just a glob, and you just I can't tell anything. So it's kind of a useless format. But another very clever, helpful graphical rendition has been developed where you use that backbone I talked about. So that those colors are showing you a completely different rendition of the tertiary structure, where the backbone is being drawn, and you're following those alpha carbon atoms, but you're using these shapes. If it's an alpha helix, you draw that helix shape. And if it is a beta strand, you draw a thick arrow. And it's a little hard to see. I don't know if you can blow it up or not, but in the inner core there are arrows that are pointing kind of right at you. So it's a little hard to see.
I can't make it bigger right now.
Okay. We'll see some I have the other graphic. We'll see some examples.
Okay.
But before we go to that, so there's an inner core of arrows pointing toward you or away from you. And they are together. They're side by side, forming this almost a complete circle. And that is a beta sheet, a curved beta sheet. And then surrounding it, there is this shell of alpha helices. And who could have guessed, who could have figured out that the sequence of amino acids would have taken on that shape, that beautiful, symmetric, harmonious shape? Is really neat. So that's why people get hooked on proteins and studying proteins, because they're beautiful and exotic and very, very clever, very difficult to predict. Can you bring up the other one, triose phosphate isomerase? So this one is you can see the beta strands a little better here. Again, we also in this one have an inner core of beta strands. You see those arrows, and they are interacting with each other. There's hydrogen bonds attracting them, and then that outer shell of alpha helices. So again, another example of a cool protein. So I just wanted to show this to give you a little bit of an idea. You can take that off now. And that's what we're talking about. And as I mentioned, predicting the protein structure and predicting the folding pathways is a very difficult problem. Making good progress on it. It seems some hope, but a very difficult problem. What about evolution? So this is now a little different problem. How did proteins evolve? Because a fundamental prediction of evolution, if evolution is true, then the species evolved. When I say the species, I mean all the species, the millions and millions, tens of millions of species, they all evolved. Nothing didn't evolve in biology. The entire biosphere evolved. Obviously, if the species evolved, the proteins, the entire molecular biology world evolved. And so that would include the DNA and everything else, but it obviously includes proteins. Proteins evolved. Now, proteins are synthesized in the cell by the ribosome and the mRNA and these actors, these different molecular machines. And the information is coming from the DNA genes. So the DNA, a gene in the DNA, not all DNA genes code for proteins. They can code for RNA, but many code for proteins. So they're called protein coding genes. And you make a copy of that and it creates a messenger RNA, as you learned in high school. And that's passed over to the ribosome, and the ribosome processes that and creates this chain of amino acids according to the DNA code. So it's this really cool protein synthesis process, cellular protein synthesis. So if you're going to evolve proteins, it's obvious that you really need to evolve not only this machinery that I just talked about that builds the proteins, but the genes, the protein coding genes in the DNA need to evolve. And then there's this chicken or the egg problem of if you evolve the gene, but you don't have the synthesis machinery, that's not going to work very well. If you evolve the machinery, then but you don't have the DNA coding gene yet, that's not going to work very well. So obviously there's a tall order here for evolution, but I'm just laying out the problem of a fundamental prediction of evolution is that proteins evolved. And that means protein coding genes in the DNA evolved. And it also means that it had to happen early in evolutionary history, because proteins are ubiquitous. They're everywhere. All the species have proteins, and they also have a lot of similar proteins. And they have this protein synthesis machinery is in the species. If it's common throughout all the species, it had to be there in the LUCA, the last universal common ancestor. So that was early in evolutionary history. So these genes, I mean, they had to be there very, very early in the last universal common ancestor. You had to have the DNA code, the protein synthesis machinery, and those genes for many of the proteins. According to evolutionary theory, other genes could have evolved later, sure. But you do have to have a substantial protein evolution process happening very early in evolutionary history. So it's a fundamental prediction of evolutionary theory. If evolution is true, then you had substantial protein evolution early in evolutionary history. So, okay, that's a little bit of background on proteins and the prediction. How does this prediction fare? Well, one problem is that for evolution here is that proteins are big. And when I say big, I don't mean physically big. Obviously, they're small. You have to have some special experiments to be able to see a protein or perceive the shape of a protein. But in terms of the number of nucleotides in a gene, so proteins are typically hundreds of amino acids. I mean, there's many that are less than 100, 70, 80, 90, 100 amino acids. Globin proteins are about 140. Histone proteins are about 100. There's many proteins that are 2, 3, 4, 500. There's even a few that are longer than that. There's a few that are longer than 1,000 amino acids. So if you have, let's just talk about 100 amino acids, a histone, that would be 300 nucleotides coding for that in the gene. Because remember, at the ribosome, each amino acid is encoded by a codon, as you learned in high school, 3 nucleotides in the messenger RNA. So whenever you have a protein, you take the number of amino acids, multiply by 3 to get the size of the protein coding gene in the DNA. So 100 amino acids in histone protein, that's 300 nucleotides in the DNA protein coding gene. That's huge. I mean, that's a lot of nucleotides a lot of mutations that have to happen to set up a coding gene that's going to create a histone. Well, maybe that's not such a big problem. Let's see. But anyway, that's just step one. Step number 2, or problem number 2, I got 10 of these problems that are going to add to each other. And these are very serious scientific problems. This is not made up stuff. Proteins are many. There are a great many proteins. When I first started getting interested in biology back when I was a little pipsqueak, they thought that there were maybe 10,000 or 15,000 different kinds of proteins, protein families. Each family has the same shape and each family consists of proteins that have the same shape, the same function. There might be some differences between them in different species. An insect species versus a fish species might have this protein that there's some differences, but they're doing the same function. They have a similar shape, so they fall into the same family. How many families are there? 10,000? 15,000? 20,000? Well, over the years, that number got bigger and bigger and bigger. We're over 100,000 now. They crossed over 100,000 a few years ago, like 2020 or so. So no, it's not 10,000. It's not 15,000. So proteins are big and they are many. Okay, so this is not a non-trivial. Now, not all of them had to evolve in early evolutionary history, but a lot of them did. So this is a non-trivial task for evolution to do this. Another problem is that proteins are taxonomically restricted or taxonomically orphaned, you might say. What do I mean by that? So you look at this tree of life, the evolutionary tree, you got all these millions and millions of species, and you pick out a protein, take a protein that you're interested in. You look into a particular species and you find a protein that's in that species. Odds are that protein is not widespread. There are some, as I mentioned, there are a number of proteins that are widespread, and they had to have been there at the LUCA, the last universal common ancestor. But actually, probably the majority of proteins are not. And so many proteins are found in one species or that species and its immediate neighbors, maybe a clade. It's not commonly found. And so what that means is you have to have a ton of protein evolution occurring all over the place. That's kind of one-off. These proteins have to evolve very, very readily. Now, when they were first discovered, this fact that there are many proteins that are taxonomically restricted, when this was first discovered, say, I don't know, 1990s or something like that, the idea was, oh, well, we probably haven't sequenced enough species yet. We haven't read off the genomes of enough species. We've just started here. We've only got, a finite set of genomes that are sequenced. So when we do more species, we'll find more copies of that one. So it's not actually taxonomically restricted. It's just a sampling bias that we've got here. It's a consequence of our sampling, and it will go away. So there's no genuine orphans. There's no genuine taxonomically restricted protein coding genes. Well, that turned out to be false. The more species, and now fast forward to 2026, we have a lot of genomes, a lot of species. The technology has, of course, improved dramatically. We can sequence the genomes of species much more rapidly. We have tons of genome data, and not only did these taxonomically restricted proteins not go away, but they actually increased. The more species that we sequenced, the more taxonomically restricted protein coding genes we found. So that's another challenge. Number 4 is that proteins are an all or nothing affair. Now, that may be a little too strong, but not much. So what I mean by all or nothing is, in evolution, what you want to, every time you look at a structure, anything, brain, the heart, bones, you want it to reveal a gradual buildup. So, gee, if we had a half of this, or a third of this, or a miniature version of this, or a simpler version of this, it would have worked pretty well. This is an interesting pathway we have, where we got a lot of things that work pretty well, and they add up to what we're looking at today. So if I look at a protein, you want to see, if I had 90% of this, it would have worked pretty well. If I had 80%, a long sequence of gradual steps that lead to this protein. And all of those steps are functional, and they're increasing in fitness. And so you have a very convincing evolutionary story to tell. That's the evolutionary narrative that you want to have. It's not the case with proteins. Proteins are like these little molecular machines, and if you cut it in half, it's not going to work. In fact, if you were to mutate just a few of the amino acids, randomly substitute in different amino acids at random locations in the protein, put in a random amino acid. By the way, we find 20 different amino acids that are used in the cells to make proteins. So there's 20 of those that are used. I mean, there's more amino acids in chemistry, but typically, and there's no rules in biology, but typically there are 20 different amino acids are used. So you randomly substitute in different amino acids into a protein, and you mutate 1 or 2, and the protein still works pretty well. But you do just a few more, and all of a sudden the function starts to degrade. And you get up to about 5%, 5 to 10% of the amino acid sequence that's randomly substituted, and your function is pretty much degraded substantially. So it's not really working. So it's not as though we don't find evidence for real nice pathways that that evolution could follow to arrive at a protein.
Okay, because the evolution is working off of random mutations, and so it needs lots of random mutations to fall upon one that might be helpful or work toward the structure you want for a protein. So in the meantime, if you've got a protein and you just mutated in a few places, it's probably going to be useless. And so the likelihood of getting the random mutations to fall upon something that's going to progress to a better protein or something is very improbable.
Going in the reverse direction. Yeah. So if you think of a graph where fitness is the vertical axis. So you want to have high fitness. And if you look at a protein, it has high fitness. It's sitting up at a high fitness. But now on the horizontal axis is the sequence. So every possible sequence is on the horizontal axis, and the vertical is fitness. And so you got this protein, like globin protein, sitting up on pretty high fitness. But now if you move around on the horizontal, so changing you're randomly mutating the sequence. So you change 1, you change 2, you change 3. So you're moving away a little bit. It falls down pretty quickly. So you realize I'm on a hill. I'm kind of close to the top of a hill. And I go down pretty quickly when I randomly mutate the sequence. So I'm kind of picturing this kind of a spike of a hill that very quickly goes down. And then way over there, there's another spike, and that's another protein. And way over there, there's another spike. So you have this kind of flat, rugged surface with occasional spikes that give you your native proteins that we find in biology. And so how would you evolve? What you need in evolution is a gradual landscape, fitness landscape, where every time you mutate, not every time, but certain mutations are going to lead to higher fitness. And you gradually can climb this hill to finally get up to a globin protein. That's what you need, and we just don't see the evidence for that. Instead, we see these spikes. Now, none of this is cast in concrete because this is a huge space. The number of possible sequences, and I'll talk about that in a second, is huge. So I'm not presenting this as hard and fast, but the science that we do understand, that we do know, is not telling the evolutionary story. So this all or nothing kind of, not a gradual buildup scenario narrative that we're getting out of the science is not helping. There's an old joke in evolutionary studies: what good is half a wing? So evolutionists have known about this all along, with wings. Like, how would you evolve a wing? Because half of a wing probably isn't going to work very well. So evolutionists said, well, it's good for warming. It's good for protection. It's good for these things. And so you're evolving these other purposes, and then it gets bigger and bigger. All of a sudden it starts to work for flying. So that's the kind of narrative you try and build up, is you've got these different steps along the way that serve some sort of a fitness purpose. Very difficult to do this in a scientific theory. We just don't see this sort of evidence.
So what good is half a protein? And you have a lot of those half a proteins running around before you ever got one that actually functions.
Right. In fact, the vast majority of just random amino acid sequences, let's say, again, back to 100, let's say you're just building random amino acid sequences, 100 amino acids long. The vast majority of them don't even fold up into some sort of a coherent tertiary structure. They'll just be kind of floating around. I mean, I'm talking about like 1 in a billion, or even worse than that. So it's a very small fraction of the sequence space even produces a foldable structure of something that's even coherent, which many functions require some sort of a coherent structure. So life is built on this very challenging property of polypeptide chains that don't fold. Okay, very rarely fold. That's the challenge that you're dealing with when you're saying, yeah, this evolved. How did this evolve? How could evolution have reconnoitered this difficult landscape? So that's the all or nothing problem. So number 5 is that proteins must have evolved rapidly. Because again, I talked about how it goes back to so many of these proteins had to evolve early in evolutionary history. We're really talking about tens of millions of years at most is what's available. Over the years, the more fossil findings we get, is just the window of time just gets shorter and shorter and shorter. And so your temporal resource is really short for pulling this off. Number 6 is, and again, that's another problem that we often see in other parts of biology. It's not just a protein evolution problem, right? Everybody's heard about the explosions in the fossil record. You go through the strata, and all of a sudden, boom, at one point in time, all these species appear. So the most famous one is the Cambrian explosion. So was it 500 million years ago or something like that? where you have all these species appearing. And it's just, well, how could it happen so quickly? You're down to tens of millions of years where these species would have had to have appeared. And there's no evidence for their, gradual evolution. And so evolutionists had to say, well, it happens real fast. And so there's this, evolution proceeds slowly and then real fast. And so you get then you get a sampling problem. We can't get those fossils. That it's so fast that they don't lay down any fossils. And so we don't see it. So you're saving the theory by, coming up with these explanations that get around the obvious evidence. So there's that. Now, another problem is the circularity involved. We touched on this earlier, but you showed this ribosome part of that video. The ribosome is this incredible machine that performs the DNA code, and it helps with the DNA code. It enacts protein synthesis. So you have the coding information being fed in the messenger RNA thread, and out the other end comes the string of amino acids. This is this cool protein synthesis problem, cellular protein synthesis. The ribosome is this fantastic machine that includes a lot of proteins. There are other machines that deliver the amino acids, and they are charged with the right amino acid by other proteins. You showed the spliceosome. Those are proteins. The DNA, reading the DNA gene to make that messenger RNA copy. There's another protein machine. So there are a huge number of proteins involved in what? Making proteins. So to make a protein, it requires proteins. That's a chicken or the egg problem. Now, how did that get solved in the real world? Like, how are you and me, how are we alive right now? How did we ever get to actually be a human being? How does a frog get to be a frog if there's this circular problem? Like, how are you going to make a protein in the beginning? When you're a zygote, how are you going to make a protein? Well, the answer is the zygote is given. These machines are bequeathed from the parents, right? So the sex cells come together, make the zygote, and right there at the formation of the zygote, you have the molecular machinery. You've got the spliceosome and the ribosome and the RNA polymerase and all these huge machines in place to do this. So, okay, that's good. I'm glad that happened. How did that all happen? So you got that doesn't help the evolutionary question.
Because you have to have a ribosome in order to build all the machines that are responsible for helping build the ribosome.
Right.
Right.
You have to have proteins to make proteins.
Yeah.
That is a fact of today's evolution, of today's science. And so you don't have this narrative this evolutionary narrative is what you need. Evolutionists had wanted the science to produce these narratives, and it just isn't doing that. You're not getting these kind of narratives out of the science. And so what evolutionists have to do is come up with these, well, it happened really fast, or, it's not visible to us. There's these heroics that happened when we weren't looking. So that's what happens. So yeah, the circularity problem. Okay, number 7, we talked about this problem of how are you going to get a protein coding gene? So let's say back to the 100 amino acid example, 300 nucleotides have to come together. Now, and that was the histone example. So you got a histone, 100 amino acids, and how exact must that be? In other words, what if I don't get it quite right? We talked about how a few mutations degrades your function, but how exactly, exact do you need to be? And therefore, how hard or difficult would it be for a random evolutionary process to get close to a protein, close enough to make it work so that natural selection could do the rest? Well, that problem has been looked at, and various studies have looked at that. Interestingly, they have come up with similar answers. Hayashi 2006 is a reference I like, but there's several others that all come up with a number around 10 to the 70th. In other words, evolution would require 10 to the 70th attempts to come up with a protein coding gene. Now, there's a couple things to say about these studies. First of all, they're all looking at easy problems. Like Hayashi 2006 looked at a part of a protein. Didn't look at the whole protein, looked at part of the protein. That allowed them to have a system that actually worked. It didn't work that well because part of the protein was randomized. So it wasn't working very well, but at least there was a function you could measure, and then you could introduce, mutations or changes and kind of measure the differences in the function. So it was an easy problem. That the other studies also had easy problems. If you get into, I talked about how there are proteins that are really big, like hundreds of amino acids, 500 amino acids. You get into those numbers, and this problem just is 10 to the 70th would be easy. That would be an easy number. 10 to the 70th is what we're finding for the easy problems.
I think topoisomerase is one of those, isn't it? It's very big and it's required, like it's in all of life that we know of. So, you've got to have it at the beginning. And
right. There's plenty of examples of big ones. Globins are 140. That's not huge, but that's still bigger. Now, I said 10 to the 70th. I want to make sure that everybody in the audience understands that. I didn't say 70. I didn't say 10 times 70. I didn't say 10 plus 70, right? I said 10 to the 70th. That's a 10 with 70 zeros after it, right? So we're talking about millions upon millions upon millions upon millions. I would have to keep on saying that for a few minutes, to get to 10 to the 70th, right? This is a very big number. So that this number is very daunting for evolutionary theory. Right? How could evolution come up with, how could it find these protein coding genes? Well, there was a paper, Dryden 2008, that looked at this problem. It was a little bit angry, a little upset with people like me. Evolution, they would call skeptics. I would say scientists, just looking at the science, but people who are doubting the ability of evolution to evolve proteins. So Dryden 2008 said, we're going to take a look at what the number of attempts that evolution actually has. How many attempts could we really put together under evolution credibly? And we're going to show that it's really, when you consider the whole Earth and all this time and everything, it's actually a huge number, and it's not a problem. So stop it, just stop. And so the number they came up with was, drum roll, 10 to the 43. So remember, we said a minute ago, 10 to the 70th, that's a 10 with 70 zeros. Well, this is 10 with 43 zeros. So that's 27 orders of magnitude short. So this was evolutionists trying to solve the problem. They were favorably inclined. They were sympathetic witnesses. They were trying to make it work. And they were making all kinds of, generous assumptions. And they came up with 10 to the 43 attempts possible, maximum. Well, we need a lot more than that. So it right there in the peer-reviewed literature, we've got this evidence. 27 orders of magnitude short. Now, be careful. I didn't say 27 times. I didn't say 27. I said 27 orders of magnitude. This is an enormous astronomical shortfall that you cannot evolve proteins according to their study. Now, they made all sorts of outrageous assumptions, by the way. One of them was you have the entire history of the Earth available to you. I just said a minute ago, you don't. You have literally a tiny fraction in geological terms, like millions or tens of millions of years. So you don't have a lot of time. They also assumed that you had the entire surface of the Earth to work with, and on that surface, it was covered with bacteria. So these bacteria were, mutating and evolving and stuff. And so they used these bacteria as kind of, the experimental platforms, the evolutionary, they were driving the evolutionary history. And it's true, the Earth does have incredible numbers of bacteria. There's no question about it. But we're not talking about that. We're talking about the evolution of proteins to begin with. Bacteria, last time I checked, have thousands and thousands of protein coding genes in them. They have proteins. They have ribosomes. They have all these things inside of the bacteria. So they are assuming the pre-existence of what they're trying to show could have evolved. It obviously is not working. So
now they might object and say, well, that's a different problem. That's the origin of life. We're not talking about that. We're talking about once life started, then evolution. So what do you say to that?
Yeah, so I mean, I would say it's a word game. Nobody, the early evolution of proteins, by the way, is not in the origin of life problem. It's these proteins have to be there early in evolutionary history, but not at the actual origin of life. But nonetheless, this is all just playing word games. Evolution has to happen. That includes the origin of all of the entities that we know of in molecular biology. If you're going to sweep these under the rug and say, well, that's a different problem, that's the origin of life, that's not part of our theory, then people realize the game that's being played here. It's just, let's be honest and show what the science tells us and what it doesn't tell us. If the science is telling us that this is not a feasible, not a plausible theory, then let's say that. And then we can also say that we think that if you still think that other things can happen, that evolution can build on that, even though it can't do this part, fine. No law against that. But let's just be honest about what the science is telling us. So that's my response to that.
Okay, and I want to get clear about what the point you're making here. You're saying that the evolution scientists wanted to see, okay, how many chances do we have to make a viable protein? And they considered covering, having the Earth covered in bacteria, and even with the Earth covered in bacteria with already functioning proteins, the chance of getting a viable protein to arise by a natural process was 10 to the 43rd?
No, it was the number of attempts that evolution could.
Oh, right. I'm sorry. Yes, the number of attempts. Okay. Yes. Yeah. They would only have 10 to the 43rd attempts.
A huge number.
Right. Okay.
Astronomically huge number that they were very impressed with. They felt like that solves the problem, but it doesn't. It's even with those very rosy assumptions, you're still far short of even the easy protein evolution problems. When you get into the hundreds of amino acids, I mean, these numbers get much bigger.
And that's only because just to give people an idea of the numbers we're talking about here, I saw a attempted calculation. Of course, it could be off a little bit, but an attempted calculation of how many total events could have happened, supposing that the narrative about the Earth being or the universe being 13.7 billion years old is correct. In that time, how many time is there for how many total events are possible? And it was only 10 to the 140th or something like that.
Yeah. I mean, these numbers are sometimes non-intuitive. Yeah. So, along these lines, Brewstead 2011 is a paper on about protein engineering. This is out of Caltech, and they said in this paper, not all protein designs are evolvable. So these are protein engineers. This is a little bit different now. This is not looking at protein evolution. They're looking at the problem of creating new proteins for industrial processes, maybe for medical processes, whatever. But they're looking at creating proteins and creating better proteins, new and better proteins. But they get into trying to find. Well, part of their job is to find new folds and find new functions, find new proteins. So they're kind of playing the evolution game, and they even use evolutionary processes. They use the word evolution in a slightly different sense. They're not mimicking the evolutionary history as as evolutionists discuss it. But they're using a selection process. They can impose their own criteria for selection. So it has nothing to do with selection in the Darwinian sense, but they call it, evolution. And so they are finding, this is really hard. And there are some proteins that are not evolvable, some protein designs that are not evolvable. So what their point was, they have to find other methods, non-evolutionary methods to find proteins. You can't just use evolutionary methods to find proteins. And so, not all protein designs are evolvable. Okay, well, and I would argue that's certainly true for real proteins, for actually probably all real proteins. But even if you said only some real proteins, that right there, is an example of what I'm talking about. That is a showstopper for this evolutionary prediction. Remember, the evolutionary prediction we're looking at is that all proteins, all natural native proteins in biology evolved. And a lot of them evolved early in evolutionary history. And so you got this problem. Okay, problem number 8, proteins, a lot of nature's proteins that we find have multiple functions. It's actually kind of unusual to have just, a single function protein. It's incredible, you find these proteins that do these different things. And so that's just another difficulty of evolving these. It places more constraints on the sequence. The sequence has to be even more clever to design this machine that can do multiple functions. Number 9 is kind of similar, but a little different here. Protein coding genes often overlap in the DNA. So we think of, in high school, Mr. Winston in your 10th grade biology class taught you that these protein coding genes are really nice. There's one, there's another, there's another. It's not that way in the real world. In real genomes, they're scattered around. They have intervening regions, and they are overlapping. And that's really crazy. So you have to design these protein coding genes that not only are going to code for one protein when you're reading one direction or in one reading frame, but code for another protein when you're reading in a different reading frame. And so there's just a lot more constraints, a lot more requirements for this protein coding gene. It contains more information that you would have to somehow evolve. So the problem, when you go back to Hayashi 2006, these other studies, they're looking at these really simple problems. It's not that simple in the real world. And then, number 10 it gets back to this chicken or the egg problem, where if you do somehow evolve a protein coding gene, just, incredible luck, you got a protein coding gene, you have to have an inheritance mechanism. You have to have, this is, passed on to the progeny somehow. That has to be already in place. Of course, we already talked about how you have to have the protein synthesis machinery in place. So there's all these major mechanisms that have to be in place. If they aren't, then so what? You did this 1 in a million, 1 in a gazillion lucky hit of finding a protein coding gene. It's just going to go away. It's going to get degraded because it's not inheritable. So a lot of issues with protein evolution. And any one of those is very serious. So I think, an evolutionist would say, well, we're still working the problem. Okay, fine, you're working the problem. But today's science has falsified that prediction that many proteins evolved in early evolution. The science contradicts that prediction. If we can't agree on that, then we're not speaking the same universal language of science, because it's very obvious, just from a theory neutral perspective. Forget about whether you're an evolutionist or not, or what you believe in, your politics and all that. Just drop all that. Just think about the science. The science is contraindicating the prediction. So we're just looking at this one prediction. Maybe evolution is true, maybe it's false. We're just looking at this one prediction. And it's clear that the science has contradicted the prediction. It falsifies that prediction. So given a false prediction now, there's something else that needs to be asked, which is actually really, really important. And that's, what's the outcome? So theories have false predictions. That's not controversial. Good theories have false predictions. Really good theories, theories that we accept as true or approximately true, often have false predictions. The difference is, so what's important is you have to look at the outcome. So it's so crucial to look at the outcome of these predictions. When you look at a good theory like Newtonian physics or something like that, that we think is approximately true, and you look at its false predictions, they have really good stories. They're really, really good stories to tell of how, yeah, there was this false prediction, but what we found, we looked into it and we found out that we had the theory a little bit wrong, and we changed the theory. We actually made it better. We simplified it. We didn't make it more complicated. We didn't say on Tuesdays it works like this. We didn't make things up. We actually were able to simplify it. It became more beautiful, and the false prediction went away. So it actually forced us to take a harder look at our theory. Or it could be we had a bad sensor, and we found out we had some bad data. And this false prediction pointed us to some particular data points. We went back and looked at that sensor. No technician had noticed, no engineer had noticed, but that sensor actually was faulty. And we found the fault when we opened it up. We didn't make this up. We didn't introduce a fault. It was there. It was a bad circuit. We fixed the circuit, redid the experiment. It all worked. And so the false prediction led to new knowledge about our experimental setup and helped us. Or, you can make up all kinds of, scenarios like this, where a false prediction is a good thing and it vindicates the theory, or at least it doesn't hurt the theory once you've figured it out. So that's what you want in science, and that's a sign of a good theory. Yeah, you're going to have false predictions, but they have a good story. In evolution, so I have 20 to 25 false predictions I'm currently analyzing, and every single one of them has bad stories. They are evolutionists solved it by adding a lot more complexity to the theory. Well, on Tuesdays it's like this. They make up ad hoc explanations because they have to. I don't mean to be critical about this. It's just that's what they had to do. Or it just isn't reconciled yet. It's just like there's no solution right now. It's just out there. It's just hanging.
But we know evolution is true.
Well, that brings up the question of why is it true? Because the empirical evidence, the false predictions are turning out to have bad stories. And we'll look at a couple right now, because what about this one in particular? Well, the stories are not good for the protein evolution, because evolutionists have been trying to fix this one and find out what's going on with this false prediction, and the outcomes aren't good. So they have admitted that this is highly unlikely, that protein evolution is highly unlikely in the literature, especially early evolution. So this is a problem. One of the explanations you will hear, I'll say a general explanation, not a specific explanation, but a general explanation is RNA did it. RNA did it. And so the solution envisioned here is that we are living in a DNA world. DNA is the holder of the genes and everything. But there was, a long, long time ago, in a far, far, far away, there was an RNA world. And so there wasn't this DNA containing all the genes. It was the RNA that contained the genes. And that was somewhere before Luca, and a lot of evolution happened in the RNA world. And then there was a transformation, and it handed over to the DNA world and took over. And that's where these proteins evolved. Well, there's a lot of problems with this. First of all, the RNA world hypothesis routinely gets shot down. If I had a nickel for every time there was another paper that's saying, oh, there's another problem with this theory. So it's not like a real obvious theory, but there is actually no empirical evidence for it. There is no relics. We don't have fossils left over. We don't have molecular fossils. We don't even have things. We can't even write a computer code that's plausible that would show how this worked. or experiment, build this up in the laboratory, an organism that is RNA based, that works and that reproduces and that evolves. We don't have that. So there's no actual thing that we would call an RNA world aside from journal papers written by evolutionists. That's where the RNA world exists, is in the literature. There's no actual real RNA world. And even if there was one, you would still face these enormous search problems that we talked about. Even in the RNA world, how are you going to find these genes? So you'll see this very commonly. Oh, the RNA world, that did it. So that's really not a very good outcome. It's not like a smoking gun kind of thing. Aha, we found a problem with this sensor. It's a theory complex. It's an ad hoc theory complexity. The complexity of evolutionary theory just skyrocketed. You've got this thing, this RNA world that's not observable. This huge thing out there. So that's a lot of complexity. Another theory that was floated was the DNA code is an enabler of protein evolution. The very structure of the DNA code is an enabler because there is an interesting structure to the DNA code. And so a lot of mutations that happen, if you were to do random substitutions in a gene, you often will preserve the physical chemical properties of the amino acids. So amino acids tend to move. When you do a mutation, you're going to tend to move in a direction that's going to give you a similar amino acid. So if the amino acid is oily, you get another oily amino acid. So, oh, that's interesting. So mutations can happen, but you'll preserve some of the key properties. And so the idea there is you can have mutations that occur that send you off into a different place in the sequence space. I talked about that graph thing with fitness on the vertical axis and horizontal as a sequence. You can take jumps. We can have mutations that move you significantly to different places, but you'll preserve these good qualities that you have. So you have this protein coding gene, and it has certain nice properties like the hydrophobic pattern, the oily amino acids. You can kind of preserve those cool properties, nice properties that help to code for a real protein. But yet you can explore space that's farther away. It's a little complicated. I know this is a little complicated to explain, but that's the basic idea. Well, it falls apart for many reasons, and you can see this in my video on false prediction number 5. So I go into this in more detail, but there's a lot of problems with this idea. I'll name just 2 of them. One is they overestimated the impact of the DNA code. It's just not true. They overestimated the impact of the DNA code by a factor of 2. And so the DNA code doesn't preserve these physical chemical properties the way they say it does. They're missing another contributing factor, and that is the sequence itself. So the sequence itself is partly doing the job here that they are ascribing to the DNA code. Another problem is that it's circular because they were acquiring a protein to begin with. A protein coding gene had to be there to begin with, just kick this method off. So that's another problem. So that's that one. I won't go into more detail on it. There's a lot more to say about it, but it doesn't work the way they think it does. Ard Lewis is another physicist who has dreamed up some of these solutions in Greenberry 2022. He was a co-author on that. Greenberry 2022. That paper was a paper about hyperdimensional bridges. Again, problems with that one. Really doesn't make sense. I won't, it's probably going to be too boring to go through all the details, but the idea was there's ways you can shortcut the process and you can get to these you can get from one protein to another by cheating, and it doesn't work. The one that's, I think, most interesting is Saakyan 2025. This was a paper that Eugene Koonin, out of Eugene Koonin's lab last summer, came out and said, look, solve the problem. We got to solve it. It's really not that big of a deal. And they built a computational tool to simulate the evolution of protein coding genes in early evolution, exactly what the problem is. and they nailed it. And it's like, yeah, it's not easy. I mean, it takes a lot of mutations, but it's certainly plausible, certainly within the feasibility of, we don't need 10 to the 43 attempts. It's not like that. It's much easier than that, maybe thousands or something, but it's not easy, but it's certainly feasible within the context of early evolution. So this was like, wow, Eugene Koonin is a very sharp guy. And here we have a paper making these incredible claims. So this must be huge. Well, it was not huge. The problem, the paper has significant problems. The most significant problem is that they have coded into their tool the target. So not exactly the target, but enough information about the target. So you're trying to take a random sequence of nucleotides and evolve it to a protein coding gene. How are you going to do that? So you're going to introduce random mutations. So you roll the dice. Okay, this nucleotide got mutated. This nucleotide got mutated. And then you're going to have a fitness assignment. If it has higher fitness, then the evolutionary process would, preserve that. It will lead to more progeny. So by natural selection. So you have to have a fitness judge. You have to have a fitness criteria, a fitness formula. So you have this random number generator. You generate these mutations and you judge them according to your fitness formula. Well, the fitness formula had the properties of a globular protein built into it. Remember I said how the alpha helices and the beta strands have a limited length. They can be 5, 10, 15 amino acids long, not much longer than that. Then they stop and there's a turn. Well, they built that into the formula. Said, oh yeah, we're just going to arbitrarily tell it that if you get longer than that, then the fitness degrades. Oh, well,
and the problem with that is that natural selection doesn't know the end game, right? Natural selection can select from what is fit right now, but it can't predict what would be fit and hold on to it. But that's what they're doing in this simulation.
Yeah. And you're starting from a random sequence. So you make one mutation from a random sequence, one mutation. You're not going to be anywhere close to a functioning protein. And yet they're going to make a judgment that, oh, there's a little bit of an improvement. Well, why was there an improvement? Because you shortened an alpha helice. You had an alpha helice that was 30 amino acids long, and you shortened it to 15 and 15. So that was better. Why was it better? I don't know. We just think we're just telling it that it's better. So there's no realism built in to actually have any kind of a realistic fitness. Another component of their fitness formula was the number of contacts that the protein forms. So when a protein folds up, it forms what are called contacts. So you fold up, and I have a visual aid here. So here is a visual aid of a globular protein. And so imagine these all stretched out, and then when they fold up, these two right here, they were far apart. Now they're close together, and that is a contact, what's called an LRC, long range contact. And so they built into their formula, their fitness formula, the number of long range contacts. The more, the better. So if you have a mutation and that brings, two things together, two distant amino acids together, then your fitness goes higher. Why? Why would that be better? We're just telling it that. Well, if you are going to evolve a protein, you have to increase the number of long-range contacts. You have to. You're going to start out with some stretched out random polypeptide chain that doesn't have any coherent structure. You have to have that become a glob with a lot of long-range contacts. So yeah, if I'm going to build a simulator, I've done this, and this is this is not rocket science. And you increase the number of long-range contacts, yeah, you'll get more long-range contacts. It'll look more like a globular protein if you do that over and over again. So that's, again, building in the target. The target is built in to your fitness formula. And as you said, you have no way of knowing that that's actually going to be an improvement. Why would that improve the fitness if you have something that's close to random, and then you got it to fold up a little bit. Would that help or not? In a LUCA, early evolution context, where it's going to be a very crude environment, there probably aren't very many, there aren't very many other species and so forth. There's no justification for any of this, except you're driving it toward the goal. Well, that's called teleology. That's teleological, where you have a final cause. So they're actually, they of course would never say that, and they didn't cast it that way. But this whole paper, the whole project, is incorporating a teleological mechanism to achieve a globular protein, the evolution of a globular protein. So it's not a realistic evolutionary process. Instead, it's teleological, and that can't be, that's strictly disallowed from evolution. So it's an internal contradiction. So that's another outcome. Remember, I talked about how important these outcomes are. When you have a false prediction, you have to look at the outcomes. Are they helpful? Are they smoking guns? Do they help the theory? Or are they things like, oh, we built in some teleology and solved the problem? We added ad hoc processes. We greatly complicated the theory. We just removed some evidence. Well, that's just bad evidence. We filtered that out because it didn't help. All those things are commonly done. I'm not making these up. These are all common responses to false predictions in evolutionary theory. Getting rid of the evidence, complicating the theory, ad hoc, theoretical components added, and actually adding teleological components, injecting kind of hidden teleology, not unacknowledged, unspoken teleology into your theory. This is not the only example of that. This is a good example of it, but there's plenty of other examples of teleology making its way into evolutionary theory. So that's false prediction number 5. And it's a significant false prediction. It's a prediction that was clearly made. It's a clear implication, a clear requirement of the theory. The theory must make this prediction. It was clearly falsified, and the outcomes are terrible. They're just really, really bad stories, not good, not the kind of outcomes you want to see. So that's it for false prediction number 5.
Great. Thank you so much for sharing that. That was a wonderful presentation. And we have some really interesting comments, so I'll bring up a few of those. Gabe says, extremely skeptical of any claim of what evolution can or cannot do because it's not mathematically possible to model both the trade space of all possible environments and all possible mutations.
Yeah, I mean, that's just pointing out kind of the difficulty with evolutionary theory to begin with. It's very difficult to model, but the science that we do know should back it up. If it's true, we should get good indications from the science. The experiments that we do, the data that we collect, they should kind of fall in line. We do have clear predictions that are made, and they clearly are falsified. So that's a real good way of judging a theory, is just looking at the history of the predictions and the outcomes associated with them.
Yeah, I think a lot of people that are watching, though, will say, oh, yeah, I've seen all those math calculations. I don't believe in all those probability calculations. Plus, we just have so much evidence for evolution. I mean, when a little problem like protein evolution comes up, hey, we don't have the answer. But since we have all this other evidence for evolution, why should we discard this theory?
The mountain of evidence argument. There's this mountain of evidence out there. And that's why analyzing the predictions is useful, because, okay, we're going to look at that evidence. Let's have a look at, let's open the kimono and let's look at that evidence. What exactly is that evidence? So you've made a lot of predictions, dozens and dozens of predictions. Let's look at how those predictions worked out. So instead of saying there's all that evidence, let's actually look at it. Let's lay it out on the table and take a history, take an accounting of that evidence. And it turns out that, yes, there is a mountain of evidence, but it isn't good. Over and over again, it contradicts the theory. So the science contradicts the theory. And remember, it's like the used car salesman. My friend told me, I lose a little bit on every sale, but I make it up on volume. And that's what we're hearing here. So, well, there's so much volume. Yes, there is a lot of volume, but you're losing on every single one. All of the evidence is contradicting the theory. So it's not a good story to tell. We can't. So often the attempts to save the theory fall back on generalities and ambiguities. And what we're talking about is specifics. Let's look at the specifics.
Right. Yep. Okay. So bring your mountains of evidence and let's look at it. Okay. Big Bad Mama says the Cambrian was preceded by many millions of years of evolution, and many of the animal phyla actually diverged during the Precambrian.
Yeah. So the fossils tell a different story. We see the abrupt appearance of dozens of forms and very, very little, very meager evidence before that of buildup. So you can always say, well, but you can't see it, but there was buildup to all those forms. It's just not in the evidence.
Okay, Miles says the first ribosomes were probably just RNA. So just along with what you said, I just thought I'd put
the
that up
RNA
there.
world. Yeah.
Okay.
It actually could be. I mean, RNA is a very important part of the ribosome, and I wouldn't be totally shocked if somebody somewhere finally someday built this halfway functioning ribosome that had only RNAs in it. That would be cool.
Miles also says spliceosomes weren't required until 2 billion years ago, at least 2 billion years after life appeared.
Right. That's why I typically don't include spliceosomes in the chicken or the egg problem, or the early evolution problem. So yeah, it's true. But that doesn't removing the spliceosome from the picture does not remedy the chicken or the egg problem. You still have a lot of proteins involved, a lot of proteins required to make proteins.
And Big Bad Mama says in the Cambrian explosion, environmental changes like rising oxygen levels, increased oceanic calcium, and a continental shelf with shallow pools may have contributed.
Yeah, the evolutionary narratives are always this might have, may have. So we can always say that the rising oxygen levels has been a thing since like the 1980s or 90s or something. They've been trying that narrative for a long time and it has not worked. But you can always throw out these narratives. This might have done it, that might have done it. The science is contraindicating that, both from the fossil record and then also from our knowledge of molecular biology and just biology in general. It's just we don't see the pathways. We don't live in a world, we don't live in a universe where unbelievable complexity just happens just by itself, just like spontaneously occurs. It's just that's not that's not what statistical mechanics is telling us, nor is the fossil record demonstrating that. So yeah, you can always throw out things if you want a theory to be true. That's good. So as a way of reckoning, we need a way of getting away from just like throwing things against the wall. What's a better way of doing theory evaluation? I'm trying to understand this theory. Let's look at the predictions. It's not the end all be all, but it's a good exercise. Look at the predictions of evolution, see what the history is, see what the outcomes are. It's very revealing.
Okay, Miles says dipeptides can act as catalysts. That's just 2 amino acids.
Yeah. So yeah, I mean, 2 amino acids can only do so much. You're not going to build a world off of dipeptides.
Okay, Mike says, hi, another good subject, Rebekah. Thanks for your support, Mike. Iron Charioteer says, aren't proteins found in the cytoplasm of the cell?
Yes, proteins are found in the cytoplasm of the cells. Proteins are found in the membrane. There are some that are transmembrane. They go all the way across. Others that are plopped into one part of the protein is embedded in the membrane, and then a lot of them are in the cytoplasm, going around doing things. A lot of them are fused with sugars or with RNA. A lot of them do, and they do all kinds of different jobs. There's dozens of different jobs that they do in the cell. So, yep, that's all true. None of that changes the fact that this prediction was falsified.
Frank says evolution could not have happened given the alleged age of the Earth, at least not the way it is presented. The theory needs to be revised to make it credible or discarded.
I mean, I appreciate the comment, Frank, but I would my pushback there is that I don't think the age of the Earth is relevant to the problem. I think you could have a lot more time, and it would still not be impossible. I don't think this is a time problem. You could have the Earth 4.5 billion years, the universe 13.7 whatever. I don't care. Give them all the billions of years that you want. These problems are very substantial, and I think beyond you don't have enough time.
Okay, Iron Charioteer says, I think we all came from viruses. I thought I'd give you a chance to respond to that.
A lot of evolutionists do say that. That is one of the going theories, that the viruses preceded the DNA world, and so that's not actually just a joke. It's actually a going idea.
And do you think we can show that that is wrong?
Yeah, I mean, we have talked about viruses. The whole virus story has fallen apart badly. So I believe this is another example. To me, this is humorous. It's just another example of when you have proponents of a theory that are so committed to the metaphysics and so want the theory, it will be true. They're not going to ever admit to the scientific problems. So instead, it's always going to be another, but there's this, the RNA world, viruses, this, that, the other thing. And this has been observed throughout the history of science. When quantum mechanics and relativity came along, there were a lot of physicists who rejected that and said, no, that's, can't be. They were stuck in Newtonian physics. And other physicists realized they're never going to change. They're just never going to change. And so the only change that's going to happen is old scientists are going to die, and then we can have change.
Okay, Chris says, give me any reason abiogenesis is impossible.
Right. So Darwin has a great passage in Origins where he talks about some of the problems, and then he says, but I have not heard anybody falsify this. And I to my mind, I'm paraphrasing this, of course, but to my mind, this just it's really hard to falsify this. And so if this can be falsified, I will just admit completely my theory is just a complete failure. But until that happens, it's true. And so this is, I'm paraphrasing again, but it's shifting the burden of proof. So if you claim that your theory is true, or is a fact, or beyond all reasonable doubt, or whatever words you want to use, then you have assumed a burden of proof. And so now we're looking at that burden of proof, and we're looking at the predictions of the theory, and we're seeing, does that substantiate, does that back up your claims, your high claims? Because evolutionists make very, very high claims. And we're finding, no, it doesn't. So now you can't come back and say, oh, but there's no reason why it's impossible. So that's shifting the burden of proof.
Okay, thank you. And Chris says, how do you calculate odds and probabilities for something like the precursor to life if you don't know what it looks like?
Yeah, it's pretty hard. It's pretty hard to do. It's a difficult problem.
Okay, so do you think that's like that a lot of those probabilities and stuff are sort of not really a good way of showing the issues?
I think it's okay because the probabilities that you find in journal papers are not this broad. They're not talking about just, what's the probability of a precursor to life. They're talking about specific problems. And this specific problem that we've created in the lab with this randomization of this part of the protein, how many tries do we need? What are the number of tries that you would need, or what's the probability? You can convert that to a probability given a certain, conditions, definitions of the conditions, and so forth. So usually the probabilities that you see in journal papers are well considered, they're thought out, and they are circumscribed. We're going to look at this very specific problem. Eugene Koonin does have that one paper where he tries to determine what's the probability of a protein synthesis type mechanism system. So it has inheritance, it has genetic information, and it has reproduction, and it has the production of the proteins. He generalizes it, so he doesn't use those terms, but he's trying to come up with what is the probability that this could have evolved in evolutionary history, such that we could have, the world that we have now. And he does come up with a probability. And so that kind of fits what Chris is saying here is, Koonin is looking at the probability of, precursor to life, and how could you do that? And it is obviously speculative what Koonin is saying in that paper, but he's taking care to try and be conservative and try and be as accurate as he can. And it's a worthwhile calculation, even if with all the caveats. By the way, his answer was, the probability was like 10 to the minus 1,015 or 1,030 or something like that. Slightly worse than 10 to the minus 1,000, which is obviously not going to happen, right? That what he showed in that paper was evolution is not possible. Now he's an evolutionist, and he believes it's true, that non-negotiable. It doesn't even consider it. It's not even on his mind. That's never a possibility. And so he used that finding to argue for a multiverse. So you have a lot more resources to work with. Remember, I said a few minutes ago, I'll give you all the time you want. I don't think time is the problem. Well, if it's infinite time, now that's different. I'm talking in terms of billions and billions and billions of years. I'm very generous with billions of years, but yeah, if you want to give yourself infinite space or infinite time, now we're in the multiverse domain. That's a different thing. So what Koonin is arguing for is, give me infinite resources, and then this becomes possible. Well, yeah, that's true. When you introduce infinity to the equation, you can take, no matter how ridiculous your idea is, it suddenly becomes a fact. It's true. So he's saying, we know evolution is true. The probability of it is insanely small. It's ridiculous. So we need to have a multiverse. And that's what it boils down to. Now, by the way, just one more thing. When I say infinity, be careful. I didn't mean literally infinity. When you talk about a multiverse, it's not infinity. It's whatever I want. So whatever I need, because I can make the multiverse as old and as many universes as I want. So I can picture a multiverse consisting of 10 to the 1,000 universes. If I find out, oh, that didn't work. Oh, okay, fine. 10 to the 100,000. Oh, that didn't work. Okay, fine. 10 to the 1 million universes. Well, that didn't work. And then you finally get to a number of 10 to the, million universes. That worked. That's all. Okay, fine. That's a multiverse. So it's whatever you want, but it isn't literally an infinity. It's just a very, very big number. Whatever size number is required to solve the problem.
And then really, this is not scientific at all, right? Because we're trying to work with the real world, right? We're trying to work with what we observe. That's what science is. So
exactly. You're getting off into that's a very good point. You're just getting off into non-science. With science, we want to figure out how nature works and how we can use nature and, work with nature for the betterment of mankind, sort of thing, right? Just kind of basics, just do science, regular science. This is all gotten into metaphysics, and it's got an agenda.
Okay, so this is I've heard this a lot, and I don't know if you're familiar, but a lot of people are really, I guess the only word I can say is just they've been misled or somehow understood. So misunderstood. So Maximus is saying 1 cell, 9 months, and then this will make a human. And so
Therefore, evolution's pretty easy.
Yes. And so do you want to address that? Like, can you talk about the misconception here?
Yeah, and it reminds me of another Ard Lewis paper. We talked about Ard Lewis with those hyperdimensional bridges a few minutes ago. He had another paper about how protein evolution isn't really that big of a deal because proteins fold so easily, and you kind of use the protein folding problem to solve protein evolution. I don't remember the exact details. I may not be representing it quite exactly, but it's that sort of an idea where I can look at nature and it works so well that evolution must be pretty easy. So you are conflating and borrowing from nature an incredible design, an incredible system that we don't fully understand, and saying that then serves as evidence that the world arose spontaneously. Obviously, those are two different things. Like, so what? I mean, so 1 cell, a zygote, turns into a human being, therefore evolution is no big deal. I mean, there's two different things. A zygote contains incredible machinery and information that if without that, you're not going to have a human, right? So it's the whole, it doesn't come for free. It's like, oh, okay, 1 cell. What do you think? It's like a random cell? Like it's just this little blob that's full of random, magnesium and, carbon and hydrogen are just shoved in there, and then it turns into a human. I mean, this is such a silly argument. It's hard to not laugh at this, and it's hard to take it seriously.
But I do want to say that, like, this is actually what people are being taught. This is like part of the whole. I mean, I've talked to so many people who are thinking like this. So we're not picking on you, Maximus, but it does show like that there is a great misconception and misleading about, what we're talking about with evolution and what we're actually observing in nature. So like Cornelius said, Dr. Hunter said, there's so much information and mechanisms already in place to make that cell develop into a human. So,
it's actually, I think this is a good example of what we touched on a minute ago. And I did a blog on yesterday of how evolution and Epicureanism has harmed science. Because the fact that a lot of people are looking at this, a lot of people are influenced by this argument, and a lot of people are saying this, hey, look, 1 cell, you got a human being. So that is a bad sign for what evolution is doing to our scientific thinking and critical thinking, and the fact, and that's Ethan Siegel had a piece out this week that I did a blog on yesterday, that in Big Think, he had the lead article in Big Think on this special issue they have, and it is really a bad sign. It's a very, very bad sign. What he's saying in that article is so non-scientific, non-empirical, it's beyond speculation. It's beyond silly. It's just like really out there. And to be writing it as fact in a public, magazine, it doesn't reflect well on him, of course, but it's a bigger problem that the bar, we've lost our ability in so many sectors, so we've lost our ability to think straight.
Can you give the gist of what he's saying that's wrong?
He was talking about the origin of life and the origin of life problem, and he boiled it down to that there's two different sorts of solutions people have been looking at. One is cell-based. How do you get a cell going? And another one is the DNA code. How do you get the DNA code going? And he was saying that, there's some new thinking out there that is looking at another solution, which is metabolic. Let's get the metabolism going. And that then will boot things up and solve the other problems. And there's a lot of problems right off the bat there. First of all, to cast the origin of life solutions into these two categories of cell-based and DNA code-based is wildly simplistic. I mean, there's way more ideas that have been thrown around in recent decades and for like 100 years. So that's just right off the bat, that's wrong. But then you throw in metabolism and say, well, here's this other solution, metabolism. What do you mean metabolism? I mean, metabolism is the digestion of nutrients in your diet, right? That's what metabolism is. These process pathways in the cell that are processing energy and so forth. So it's that's a concept almost. So again, these are a set of pathways. How could that then solve the origin of life problem? You have to have something a lot more concrete than that. So it's getting more and more vague and ambiguous. Okay, maybe there's more to it. Let's read the article. Let's read the papers he's referencing. Maybe there's more to it. Well, it just gets worse. And he has some passages that it's hard to exaggerate how ludicrous the theory is that he is the science that he is proposing. He's making statements that just have no empirical backing. Like, where did you get this from? There's no citations. They're really laughing. You have to kind of watch the video, I think, because it's hard. It's hard to recreate.
Yeah.
So.
Okay. Theories of Creation says, great show. Thank you. And Dave says, some people seem to have trouble understanding that because we don't yet know a particular evolutionary pathway doesn't mean one can't have existed.
Right. So the argument from ignorance, or you're being too credulous. Just because we don't know it all, that doesn't mean we're there. It's impossible. No one has said that, actually. So remember, what we're doing here is we're looking at predictions of the theory, predictions that are unambiguous, that proponents of the theory made, and that have unambiguously been falsified, and that you have responses to outcomes. And we're looking at those outcomes. And a false prediction can be okay. It can be perfectly fine. But in the case of the predictions that evolution makes, it's not. They're not good outcomes. So it's not the case of just being incredulous or credulous and just saying, oh, we don't fully understand this yet, so therefore it must be impossible. No, you're not reckoning with the problem. You're not genuinely reckoning with the science and what the science is actually saying. It's much, much more serious than how you're conveying it, how you're characterizing it.
Yes, we're looking at what we do know, right?
Yeah.
So Drake says that there is a mountain of evidence for abiogenesis because we have observed organic molecules form spontaneously in prebiotic conditions. Yeah,
so it's a little difficult to see if this is being sarcastic or not.
No, not being sarcastic.
Okay, because this is pretty bad. Yeah, it's that the formation of organic molecules such as an amino acid or a nucleotide or a methane molecule or some relatively simple organic molecule. You can find that on meteorites or spontaneously formed in a test tube or something. That doesn't solve the origin of life. So a methane or a nucleotide doesn't equal a bacteria, no matter how simple you make bacteria. In fact, they tried to do that. One of the experiments they performed about 10 years ago or so was to take the simplest cell you could find. It had about 1,000 genes, and let's start removing genes one at a time and see how much can we simplify? What is the simplest life form? And it didn't work. They got down to about 400, 450, 500 genes, and that was kind of the end of the line. They were hoping to, get it down to something a lot smaller than that. Something that, if you get this thing down to 10 genes or 5 genes or something, what are the essential genes? Well, it was like 500 genes. So you're a long way away from life when you form a nucleotide or an amino acid or something like that. Hydrocarbon chain. Tell me one thing that contradicts evolution. One thing. There's this one thing. Yeah. So what you need to do is look at the science. I would say the 20 false predictions are a pretty good start. If you don't know of anything that contradicts evolution, then you're not understanding either the theory or the science. So in this false prediction number 5 that we did today, we're looking at the prediction that proteins must evolve, must be evolvable, and especially in the early phases of evolution, early stages. And that just didn't turn out to be true. The science contradicts that. The science shows clearly, crystal clear, it's not going to happen. Now, future science can change that. Maybe that's going to change. I'm talking about current science. So if you want one thing that contradicts evolution, watch this video or read or watch my video on false prediction number 5 from my channel, and you will see, you will understand that the one thing that contradicts evolution that we're talking about today is protein evolution, the prediction that proteins must evolve, must have evolved, must be evolvable.
And this is a good time to let you know and the audience know that if you want more of evolution's false predictions, Dr. Hunter has a great summary of false predictions that evolution has made. And I've linked that paper in the description of this video. So you can access that for free and read for yourself a very concise summary of many false predictions that evolution has made. So check that out.
Let me butt in. I'm very sorry, but I am working on that. It's it's under construction right now.
Okay.
And so the link, I'm sorry about this. The link doesn't work right now.
Okay.
It is out. People have gotten it. It is out there and everything. But I have a bunch of videos. You can go to my channel and watch the videos on false predictions. So it is available to you. But the hard copy that Rebekah, I provided to Rebekah, the link to, is right now, it is disconnected. I'm sorry about that. I should have told you that.
Are you okay with me giving them the previous thing if they email me?
Yeah, okay. Chris or anyone, if you contact Rebekah and say, hey, could I have a look at this thing? Feel free to send that to, knowing that it's under construction, but it's a good, perfectly good document of 23 or something false predictions. And so, Chris, if you're genuinely interested, like I was at one time many years ago, I was an evolutionist and I got intrigued at what are the evidences for this theory? Why is it a fact? If you're genuinely intrigued and you really want to know, as opposed to if you are committed to evolution, then I wouldn't, waste your time. If you're not going to change your mind, then, I would drop it. But if you're genuinely, honestly interested in learning the science and the theory of evolution and what the theory of evolution really is, because it's not from empirical science. The theory of evolution, the fact of evolution was never motivated by and was never justified by the science. That just never happened. It didn't happen in Darwin's book. It didn't happen with the Epicureans 2,000 years ago, and it isn't happening today with the scientific evidence in the literature. It doesn't. The fact of evolution never came from the science, and there are a bunch of arguments that evolutionists do provide for the fact of evolution. They're not shy about it, and if you look at those arguments, you'll see it's not science. Those arguments are not coming from science. They do use scientific lingo, but the key premises are not scientific. They're metaphysical.
Yeah, they are religious. And this actually this comment comes along at a great time for that. Iron Charioteer says, religion does not promote critical thinking, though. Well, it depends what your religion is, but evolution is it's upheld by a lot of religious thought, and believe me, they don't want you to do critical thinking.
Yeah. So I would agree with Iron Chariot that, in the case of evolution, you don't see critical thinking.
And so Drake says the evidence for macroevolution is homologous structures, genetic similarities, rapid artificial selection showing pathways to see massive phenotypic change, radiometric dating, etc. I love this comment because this is actually giving us some substance to work with. So I'd love to hear your response on this, Dr. Hunter.
Yeah, I agree. Great comment. Good to see some actual evidence being cited. Homologous structures. I'll give you an example of a homologous structure. There is a homologous structure. It not only is a homologous structure, but it is one that is probably the most commonly cited as a proof text of evolution. It was cited by Darwin. It was much talked about back then, but still today you can pick up, biology textbooks from your high school, college, and you'll probably have an image there of the pentadactyl pattern that you can see in the mirror on your hands and your feet. And it is thought to be a very powerful proof or confirmation of evolution because you find it in so many species. So over and over and over in all these different species. Now, the pattern itself does change a lot. It looks very, very different in a bat than in a human or a fish or a horse. They look very, very different. But a trained anatomist can look at it and say, yeah, there you see the evidence for the pentadactyl pattern. So there you are, homologous structures. You are citing evidence for evolution that is right there, cited by the major leading evolutionists who are publishing professors, textbooks, popular books, magazine articles, videos. That's a very common one. It's not just the pentadactyl structure. Of course, there are many other examples of homologous structures. So there you go. Now, the question is why is it so strong evidence? So you could argue that, okay, evolution predicts similarities. That's a similarity. Therefore, you have a confirmed prediction. Okay, great, wonderful. But a confirmed prediction, the flat Earth makes confirmed predictions. Geocentrism makes confirmed predictions. All kinds of theories that we would take as false make false predictions. So if we have a confirmed prediction, okay, what are we going to do with that? Why is it particularly strong? Well, this is cited as much, much more than just a confirmed prediction. It is a literal proof. It is just, wow, that this thing is just a proof. And why is that? How do we get from a confirmed prediction to like slam dunk? That's a big jump. And the way that was done, and this is a perfectly valid way of arguing. So I'm not saying this is illogical. It's perfectly valid. And Darwin made this argument very powerfully, is that if you have a similarity like this, like the pentadactyl pattern in these different species, it can't be optimized. This must not be the optimal structure because it's used for grasping, swimming, flying in the bat, walking, crawling, digging, the mole, digging, So it's used for like a half a dozen different purposes. And if you were to design something for such a different purpose, you certainly would come up with a different design, right? And John Owen, leading anatomist in Darwin's time, pointed this out as much as anybody. Like, this is not working. This is not the idea that Cuvier and others pushed, and many evolutionists and natural theologians sometimes, not always, but sometimes advocated, was that everything we find in the world is optimally designed. It's adapted, perfectly adapted for its environment. Which of course, anyone who believes in a all-powerful God might be amenable to that, right? That might be like, yeah, that makes sense. Like God made the world, and so everything's gonna be perfect. Yeah, perfectly adapted. That makes sense. And so that's what we see, right? Well, Darwin and Owen and others were saying, well, no, actually, the pentadactyl pattern shows that you have a repeated design for different functions. That right there tells you that it's not optimal. It's not the best that it could be. Okay, so what does that mean? It means that God did not design it, intend for it, or create it. Well, then how did it come about if God didn't do it? It came about by natural laws, natural processes. It came about by itself, so to speak. It spontaneously arose by some sort of natural process. Oh, what's that called? We call it evolution. Oh, does that mean like mutations? Well, that's one possibility. There's all kinds of possibilities, but they have to be natural. Why does that have to be natural? Because we just showed you that God wouldn't have done it that way, because these aren't optimal. Oh, I get it. They're not the best that they could be. So that's an argument from disutility. Disutility, or the argument more generally, theological utilitarianism, is the category. That is one of the most powerful arguments for evolution, both in Darwin's day and today. Things that don't work quite right. Now, I said that this is a perfectly logical argument, and it is a strong, powerful argument. It's why somebody would put it into a comment, not as a confirmed prediction, but as a proof, to a powerful evidence. It moves from being just a confirmation of a prediction to a powerful evidence. This is very powerful, because you're not saying this is a confirmed prediction. You're saying it is a disconfirmed prediction. It is a falsification of what? The alternative. In science, it's way more powerful to falsify the alternatives than to try and argue for the theory that you're a proponent of, because arguing for a theory is so difficult in science, because confirmed predictions only get you so far. But if you can reject something, oh, okay, get rid of those other ideas. I'm the only one left. That's the idea here. It's this counter arguing, and that's what this is. Now, there are, by the way, some problems. I said it was a perfectly valid, logical way of arguing. However, now let me point out some problems with it. And the first problem I'll point out is that it's difficult to judge what is optimal and what is the best adapted. Remember, I said the bat wing, yes, it's a pentadactyl pattern. However, it's like radically different in terms of the abstract, the ratios, the geometric ratios, and so forth. So yes, you can pick out a pentadactyl pattern, but it looks very, very different than, say, the human hand, or the horse hoof, or whatever. So it's measuring what is optimal and what isn't in biology is really hard. I'm not arguing that it is optimal. That's not my point. I don't care if it's optimal or not. I'm just pointing out that the assumption that, oh, it's the same pattern repeated over again, therefore it can't be optimal, is not a done deal. It's not proven. I would say it's maybe a reasonable idea. It's certainly a reasonable idea, but you can't really bank on it. So that's problem number one. There's two more problems. Okay, so they got three problems. The second problem is the observable is actually false. So remember, the observable is the pentadactyl pattern dominates it. All you find is the pentadactyl pattern. Well, that's what they thought, except when you looked under the hood, there's actually other patterns as well in biology. You can find animals that have 6 digits, 7 digits, 4 digits, and all of a sudden, this got more complicated. And that's another one of my false predictions, where the pentadactyl pattern failed. Homologies in general fail, and they fail hard. They fail very badly. The entire argument, when you actually look at the anatomy of the species, all of a sudden you find, yeah, there was this general pattern that caught our eye, but guess what? It's riddled with problems. And I'm not talking about minor problems. I'm not talking about problems that can be solved easily under evolutionary theory. Like, yeah, you've got some exceptions to the pentadactyl pattern, but they're all in this one other clade here. They're isolated geographically and taxonomically. And yeah, there's a mutation that happens, and you went from 5 to 6, and then it just was 6 on the whole way. Something kind of simple and straightforward. No, not at all. It's all over the map. And so from an evolutionary perspective, they had to end up hypothesizing all kinds of losses and reacquisitions of the pentadactyl pattern and the arising of other patterns here and then there and then there. It is a very scattered diagram. In the evolutionary tree, you have these, it's non-parsimonious. It is not a parsimonious explanation. You had to now remember I talked about how the outcomes are so important? The pentadactyl pattern prediction failed and failed badly, and the outcome is a non-parsimonious explanation. So theory complexity skyrocketed. And so you have all these mutations happening here, there, and losses and reacquisitions of these patterns. So if you're going to make that argument that this proof text, this proves evolution, then by modus tollens, you have to now agree that the falsification of the prediction falsifies evolution. Remember, if you had merely said, yeah, this is just a true prediction that came out true, not that big of a deal, because true predictions aren't really that big of a deal in science, but it is a true prediction. I wanted to point that out. And then if I told you it was a false prediction, you say, oh, okay, well, it wasn't that big of a deal to begin with. But when you point it out, as evolutionists always do, as this slam dunk, powerful proof text of evolution, then we're in a different ball game. Now we're in a case where if it falsified, this is a hard prediction. It's rock hard. You cannot sustain the falsification of this prediction now because you made it a big deal. Well, now that it's been falsified, you have to agree that evolution is false by modus tollens, or else now you are illogical. And now you are taking an illogical stance. Remember I said this is a logical argument to begin with? Okay, so evolution now is false. You have to agree that it's false. Okay, that's the problem number 2. Now, the third problem. Remember we said if God had created the world, then God's all powerful, all knowing, all good and all that. So he's going to make these species optimally adapted for their conditions in which they are found. That's the key phrase. Designs are best adapted for the conditions in which they are found. That's Cuvier's wording. And that's the wording that everyone adopted. It's optimum. It's best of all worlds. Where did that come from? Well, if we look at the Bible, it doesn't say that. In fact, it's very clear that that's not always the case, at least according to our judgments of what is good and bad. Functional adaptability, the best way that a design could be made. No, God says, no, I didn't do it that way. It's right there in the Bible. You can look at Job 39:17, for example. It's, no, I did it this other way. It's not as good as it could be. And Job 39:17 actually provides the reason. That's just really cool about this passage. It doesn't say because of the fall, or because of natural processes, or your sin, or because I am a distant sovereign creator off meditating on Mount Olympus, and I don't really care about the world that much. You guys are, deal with it. It doesn't say that. It says, because God did not do it that way. I deprived the ostrich of wisdom, therefore that's the way it is. I did it that way. Total taking credit for it, right? God takes credit. He clearly, unambiguously states, I did it that way. And it's not what you like. It's not theological utilitarianism. So the argument that you're providing here, Drake, this homologous structures, falls apart for multiple reasons. There's multiple problems with it, even though at first glance it kind of looks, yeah, okay, this is a logical argument and everything, and got good evidence and everything. No, it falls apart. So that's what you need to understand. That people need to understand this. This is the story of evolution is not just science. It's not just history. It's not just philosophy. It's not just theology. It's all these together. It's the overlap of all these things. That's why it's such an interesting topic. And once you start to get to grasp this, you'll start to open your mind and open your blinders and realize, oh, I've been brainwashed and I got fooled. And a lot of other people got fooled. And I think my teachers were fooled. And there's a lot of depth here. It's a very interesting subject, and you can learn a lot about the world. So when I teach this class, I make it clear to the students, this isn't just one thing that you're learning here in this class. And we're not just going to talk about one thing. This is the philosophy of science, yes, but there's all these other things wrapped up. And you're going to open your eyes more, and you start to realize there's a lot more going on in the world than the stuff you've been told.
Yeah. Now, this part of the comment actually goes with another comment. So the part about rapid artificial selection showing pathways to see massive phenotypic change. But it kind of goes along with this. Junior says, ask him if he is aware of what evolutionists are calling contemporary evolution, reports in the literature of physical changes in species, even as fast as a few generations. I guess artificial is different than this, but I kind of wanted you to address this one because this is, something that comes up a lot.
Yeah, I definitely am well aware of rapid evolution, what I would call rapid adaptation. I wouldn't call it rapid evolution. But when you say physical changes in species, even as fast as a few generations, yeah, for sure. I'm definitely aware. So ask him if he's aware. Yes, I'm well aware of that. I would call that adaptation, and I would call it directed or intelligent adaptation, because it is within a generation or a few generations. And so, in other words, it's not by random mutation and natural selection. So RMNS, random mutation, natural selection, that is kind of the favored, mechanism for evolution. And that's how it has to work. It has to be undirected. This is directed. And so this is another contradiction of the predictions of evolution. I've done several videos on this, and I've also discussed it in my false predictions paper that Rebekah can send to you. But yeah, the unfortunate thing about those examples, Junior Silva, that you're talking about there, is that they are often labeled as evolution. And so in other words, evolutionists are taking credit for that. So saying, yeah, look, here's an example of evolution. So no, it's not evolution. In fact, this is a problem for evolution, because what you're seeing there is species have built-in directed adaptation mechanisms. In other words, you can take a species and expose it to a challenging environment, which maybe it has never seen. It may be never in the history of the world was that species exposed to that challenge, or at least that we know of. And yet it adapts. It adapts to it. And that's really cool. And we see all kinds of examples of this, like you say, within a generation or a few generations. And a few, So, a lot of individuals in the population are making a change quickly. Instead, the evolutionary story is one individual gets a random mutation that helps, and then slowly that propagates throughout the population over a long time period. That's the evolutionary narrative. Random mutation, slowly propagating by what we call natural. We call that process natural selection. That's not what's happening. That's not what we're observing. We're observing a rapid change happening in parallel, simultaneously across the population in multiple individuals, because they have built-in directed adaptation capability. Where did that come from? That's not predicted by evolution. In fact, evolutionists fought that. They fought that. This wasn't just they didn't predict it. No, they fought it. They denied it. And when it became undeniable, they said, well, but it doesn't really matter. No, it does matter. Well, it doesn't really last. It's not robust. No, actually, it is robust. Well, okay, evolution created it. Well, no, you can't have your cake and eat it too like that. You can't fight it. It totally demolishes your theory. And then at the very last minute, you say, well, we just created it. Well, the reason why it doesn't work for evolution is because it wouldn't be selected for. Well, there's two reasons. It's too complicated to evolve by random mutation. So there's that. But even if you somehow luckily constructed this system of rapid, intelligent, directed adaptation, it wouldn't be selected for because that environment is not obtained right now. That's a future environment. So organisms have this capability to respond intelligently to an environment that isn't here right now. And so if you evolved that, it would just simply mutate away. It would be degraded. There would be no selection for it. There'd be no purifying selection. It wouldn't happen. You're not going to evolve it. So it demolishes evolutionary theory. It's just another example of how the science that we do know contradicts the theory.
Thank you. And Junior did want you to know that he's not an evolutionist. So he was asking you that question, as someone who is on your side.
That would help the narrative. Well, thank you, Junior.
And we do need to close up because it's already past 2 hours. But Drake, we had his comment up before, and he said he never claimed homology alone was a slam dunk proof of evolution. You strawmanned me in real time and ignored most of my comment by focusing only on homology. Okay. So I don't think that was your intention. I took the comment down and moved on. But let's put the comment back up, and I'll let you respond.
Yeah, I think your point about it's 2 hours in and everything's like that. I hope you get the point, Drake. So first of all, I apologize for dwelling on the first one where your argument may have been, hey, look, I've got all these evidences and no one of them is all that great, but I've got them all working for me here. And so if you knock just one of them down, so what? I've still got all these other evidences. So I'll acknowledge that was your point, and I'm sorry about not dwelling on the first one. So good point well taken. However, you're going to have problems on the other ones also. So it's not, I want you to, if you can open your mind and realize it's not as though, a guy that comes along like me and points out a problem, that's just one problem. That's just not it. The evidences and arguments for evolution are contradicted by the science, but confirmed by the religion. So these are all inspired by the religion. I mean, radiometric data. I mean, I not gonna, each one of these would take a while. You can look through my my channel, my papers that I've written, my books that I've written, or the paper that Rebekah could provide to you, if you want more details. But it would take a long time to go through these. But I hope you're not. I want to hope that you and others are not thinking, yeah, there's just so much evidence for evolution. Yeah, you can knock down, you can pick out a weak point. No, that's not it. We're talking about a theory that claims the world arose by itself. That is a heroic theory. You need to have evidence for that. Why should we believe it's a fact? Then when we look at the evidences, like the ones you're pointing out here, they don't work. They have problems. Believe me, every single one that you have here has problems. These are not good evidences for the theory of evolution. I talked to the first one. I could go on and on and on about all the others. It would take a long time to give the full story. But there's no meat there. It's what you find is problem after problem after problem after problem. There are serious problems from a scientific perspective. And when you look at the powerful arguments for evolution, all the really powerful evidences that are put forth, they are relying on, they're incorporating and motivated by these theological premises. And that kind of makes sense. I mean, it kind of makes sense that you have a theory that really is out there, like, okay, the world arose by itself spontaneously, really? And that's a fact? Like, okay, where'd you get that from? Oh, I see. You got it from Epicureanism and theologyism and all kinds of isms that I don't believe in. So, I'm not affected by this.
Thank you so much, Dr. Hunter. This has been really great. And thank you for the audience. Thank you for tuning in. And if you want to get in contact with me, you can get in contact with me at examiningorigins@gmail.com. And I'll give you Dr. Hunter's last, update for his false predictions paper. And then he's working on some edits to that. So hopefully it'll be up and running again soon. But yeah, thank you all. And I hope everyone has a great day. God bless you.